Hello, thank you for showing interest in my poster. My name is Kawtar. I am a PhD student in Dr. Li Dang's group in Munich, Germany. The project depicted in this poster is focused on the method called differential RNA sequencing, as opposed to RNA sequencing, which allows the study of gene expression changes. Differential RNA sequencing is a high-throughput screening technique that enables the enrichment of primary transcripts, which allows the differentiation between primary start sites and processing sites. And this leads to the annotation of various transcriptomic and regulatory sites, such as transcription start sites, operon structures, non-coding RNA, and ribosome binding sites, which in itself allows the understanding of RNA maturation pathways. The experimental workflow starts with infecting bacteria with bacteriophages, then taking samples at specific time points, extracting the total RNA, and then the enzymatic treatment with the terminator exonuclease or the TEX enzyme that digests the processed RNA transcripts, which indirectly enriches the newly transcribed non-processed transcripts, which we can then analyze for transcriptome annotation. So in this project, we focus on interactions between a baumannii and its lytic phage HMGU1 in presence of select antibiotics in order to study the potential synergy between phages and antibiotics. In the presence of antibiotics, the phage transcriptome looks different. Rifampicin is an antibiotic that inhibits bacterial DNA-dependent RNA synthesis by inhibiting bacterial DNA-dependent RNA polymerase, and tigecycline blocks the interactions with the ribosomal subunit 30S. The effect of the TEX enzyme can be visualized in the enrichment patterns at potential transcription start sites, while the effects of antibiotics are reflected in different expression patterns that are either limited to the middle genes in the case of rifampicin, as opposed to a more overarching expression throughout the genome in the case of tigecycline. For the analysis of the data, we use bioinformatic tools like Redemption and Analgesic, which were specifically developed to generate annotations of genomes using a genetic algorithm and machine learning approaches that are trained based on user-curated, manually detected transcription start site prediction. So the input we would give the tools is the genome sequences, and we would apply different packages like the transcription start site, processing site predictors, or terminator predictors, which would then give us the carefully calculated and detected annotations of processing sites, transcription start sites, UTRs, terminators, and transcripts. From our analysis of the differential RNA-seq data, we could conclude the number of uniquely aligned reads mapped to the bacterial and phage genomes, while the transcription start site annotation of the bacteria genome provided an insight on the overlap between the primary, secondary, internal, antisense, and orphan transcription start sites. Our preliminary results showed that different genes are differentially expressed in each treatment. However, the expression of hypothetical proteins with unknown functions is prevalent in all treatments. Preliminary results suggest that while both antibiotics interfere with phage replication, rifampicin acts as a more efficient downregulator of phage structural proteins, while presence of tigecycline upregulates the expression of predominantly unknown proteins. Combination treatments of select phages and antibiotics have been shown to have synergistic antimicrobial effects. On the other hand, fewer research has been dedicated to the effect of these antibiotics on phage replication and synthesis. Our preliminary results suggest that HMGU1 phages behave differently in the presence of either rifampicin or tigecycline, with different genes being expressed in response to the presence of either antibiotic. Rifampicin seems to downregulate structural protein synthesis, while presence of tigecycline seems to upregulate the expression of mostly hypothetical proteins of unknown function. Our research is focused on studying the effects of synergistic antibiotics on phage production, as well as the characterization of the regulatory potential of unknown phage proteins, in order to enhance combination treatments against multi-drug resistant bacteria. Thank you for your attention.